right, guys, welcome back. So this is the second video for this week. It's on the 4 different biomolecules. You should have it in your OneNote Unit 1 called Biomolecules. So basically what this video is going to be is sort of a deep dive into each of the 4 biomolecules, which are carbohydrates, lipids, proteins, and nucleic acids. And I'm going to do them in that exact order and just kind of talk about what they do and why they matter. So we're going to start with carbohydrates. Um, they're going to be really important for the lab that we're doing this week, but they're also just my favorite because they're delicious. So we'll start with that one. All right. So when we talk about carbohydrates, the functions of them are that they are short-term energy source. And they also help with structure and support in cells, especially the larger carbohydrates. But most importantly for us is going to be this fact that they are quick energy. This is why if you're going to like run a race or something, you eat carbs the night before because they're the quickest energy source. All right. So when we talk about carbs, there are 3 different categories of them. The first one is monosaccharides, and then we kind of talked about this in the last video. But monosaccharide means one sugar. And these are also known as simple sugars. And we use them for quick energy sources. Um, the example of this, the main one that we're going to care about in this class for like the whole entire year, is glucose. Glucose is a monosaccharide whose formula is C6, 6 carbons, H12, 12 hydrogens, O6, 6 oxygens. And what's kind of interesting about glucose is that it can either be a ring structure or it can be a linear structure. So I'm going to put this here, ring or a line. Whether it's shaped like a ring or a line is going to really affect its function. So in biology, we're going to talk about this a ton of times this year, that the shape of things really is super important for their ability to do their job. And that's true about this sugar. It's going to be true about proteins and a ton of other things. So when you have things that are the same atoms but different structures, we call those isomers. And it's actually one word. So it's isomer. But I wanted to break it down to kind of tell you what it means. So iso always means same, and mer is molecule. So basically what this word means is that it's the same molecule, same atoms, same number of atoms. They're just arranged a little bit differently. And that's what's true of glucose in the ring shape or in the line shape. So I'll show you a picture of the 2 glucoses. Here they are. So they're monosaccharides, both of them. But they're both glucose. So I'll just say glucose here, glucose here. And these are just structural isomers. Because they're the same atoms, but they're a different structure. Both are important. All right, so now what we're going to do is we're going to start to use our monosaccharides to build larger carbohydrates. So if I come down here, the second type of carbs that we're going to talk about is a disaccharide, which is 2 monosaccharides bound together And when you make a disaccharide, you're going to use covalent bonds. And when you're using a covalent bond in between two monosaccharides or multiple monosaccharides, it's called a glycosidic linkage. Really the same as any other covalent bond, but if it's a bond for a carb, that is what we call it, glycosidic. And then just remember from the last video that glyco means sugar or carb. So anytime you see that glyco, you can just have sort of a little hint that it is related to a sugar. So if we're going to talk about an example of a disaccharide, one is sucrose, which sucrose is actually the sugar that like you would use to bake a cake. It's the one that like you buy at the store. Sucrose is, so I'll just say that this is like table sugar. Sucrose is a glucose bound together with a fructose. And you can see here that glucose and fructose actually look super similar. Um, they're just really, really, really slightly different. And what's actually different about them is the arrangement of the molecules. So, I'm sorry, the atoms. And fructose is also the sugar that's found in fruit. So just remember too that ose, I'll put it over here, means that you're talking about a carb. It's an easy way to remember if the word ends with ose. All right, so now we're going to talk about the biggest type of carbs, which is a polysaccharide. So come down here to number 3. Polysaccharides are many monosaccharides, which remember poly is many. So I'm talking about like hundreds to thousands bound together covalently. And these are also called complex carbs or starches. I'm going to talk to you about 4 different polysaccharides and what they do. The first one that we're going to talk about is actually animal starch, which might be something that you've never really thought about before, but we actually do store starches in our bodies to use later. The starch that we store is called glycogen. So there's glyco again, coming back up. And your glycogen is actually stored in your liver. Um, so starches are going to be slightly longer term energy than a simple sugar would be, but still not even close to really being long term sustainable. If you stopped eating, all of the glycogen in your body and your liver would last you about 1 day. So it's really not a very long time at all. The other interesting thing about glycogen is that when your blood sugar starts to get really low, glycogen is broken down and released as glucose because that's what it's made of, is just a bunch of little glucose molecules bound together. And then when your blood sugar gets high, I'll put this down here. glycogen is stored to remove glucose from blood. And this is the job of the molecule called insulin, which is a protein. We'll talk about it in a minute, but it's really important for maintaining your blood sugar. And if you have diabetes or you know anyone that has diabetes, then you know how important it is to actually maintain a stable blood sugar to have homeostasis in your blood. And it's your insulin that is responsible for making sure that your glycogen is broken down and released if you need to have more sugar, or um, stored if you have too much sugar in your blood. Okay, so the second type of polysaccharide that we're going to talk about is plant starch. And plant starch is called amylose or amylopectin. Um, an example of this, like foods that contain this, are going to be your breads, potatoes, pasta. Um, and then when you eat these things, your body breaks down starch using an enzyme, which enzymes are proteins. But the enzyme is actually called amylase because it goes with amylose in your saliva. So starches are actually digested in your mouth. A lot of other things are digested in your stomach, but not starches. And that's the reason why if you've ever like left a cracker on your tongue for a long time, it eventually will start to taste sweet. And it's because the starches are literally getting broken down into the simple sugars. And you can taste that happening. Kind of gross because it gets super mushy, but it's also science at work. So there you go. Both animal starch and plant starch are um, energy storage carbs. So I'm going to label them. I guess I'll just go out here. So this is energy storage polysaccharides, but they're still considered to be pretty short term in terms of energy. The last two we're going to talk about are slightly different. So we'll go over here to talk about it. All right, so polysaccharide number 3 is called cellulose. So again, ose. Cellulose forms the cell walls of plants. And what's kind of interesting about cellulose is that you actually cannot break it down in your body. And so it is fiber. Plant starch, you are able to break down, but cellulose is a polysaccharide in plants that you actually can't break down. Because it makes up the cell walls, it's very sturdy um, and rigid. So really strong. It forms in like long chains of glucose. And then the last carbohydrate that we're going to talk about that's a polysaccharide is called chitin, which is pronounced like kite in. It looks like chitin. It's not, it's chitin. And this is found in the exoskeletons of arthropods. Arthropods are things like insects, lobsters, crabs, scorpions. It's actually arthropods is the most diverse phylum of animal life, which is pretty interesting. And it's also found in fungi. We don't really use chitin, but other animals and organisms definitely do. And it's structural, it's making up their skeleton. So these two, cellulose and chitin, they are structural carbs, structural polysaccharides. So they're not really an energy source. They're used for other purposes besides energy. All right, and that's it for the carbs. So you have your monosaccharides, your disaccharides, and your polysaccharides, going from simple up to complex. All right, let's keep going. Next one is lipids. So we're going to come down here to talk about that. Lipids are your fats, oils, waxes, and their functions are going to be long-term energy, as well as insulation, which you might know has to do with retaining heat. So a good example of a lipid doing this is whale blubber, which is fat. Lipids, because they're fats, often get like a bad reputation because people are like, "Oh, fats are super bad for you." That's not really true. There are some fats that are really bad for you in terms of like your cardiovascular heart health, but they're actually really important, and your body has to have some levels of fats in order to function. So it's really important for a lot of stuff. Don't think that they're always bad. Lipids are hydrocarbons, which means that they're made of only hydrogen and carbon. And because they're only made of hydrogen and carbon, they are nonpolar, which means that they resist water because water's polar. And we call that hydrophobic. So again, hydro means water, and phobic is fear. If you have a phobia, you're afraid. So you can think about them as literally being like afraid of water. Carbs, I should have mentioned this before, but carbs actually contain, I'll go up here and put it with them. So carbs, elements are carbon, hydrogen, and oxygen. So that oxygen makes them a little more polar and a little more willing to react with water. But if I go back down here to lipids, they are hydrophobic. They hate water, which you've probably noticed if you've cooked with oil. So examples of fats. We already talked about fats, waxes, and oils, but I'll also put on here steroids, cholesterol, which there are good and bad versions of cholesterol. Um, Also hormones. Estrogen and testosterone are actually lipid-based hormones, and phospholipids. Phospholipids look like little jellyfish. They have a phosphate head and they have lipid tails. Okay. Three other examples that I want to talk about quickly because they're related to your health in a way. are saturated fats, unsaturated fats, and trans fats. So the first one is a saturated fat. Here's a little picture of it. What it's showing you is that there's only single bonds. Let's say single bonds only. And this is important because there's only single bonds, which allows the maximum number of hydrogens to attach to the carbons. So that's what it's saturated with, is hydrogens. Because it's saturated, there's not a lot of space in between the hydrogens, and that creates things that are most likely to be a solid, just because everything is packed really tightly. So an example of a saturated fat that you're familiar with is butter. Saturated fats are typically solids at room temp because they're packed with maximum number of hydrogens. Okay. All right. So unsaturated fats are a little different. They have a double bond. So you can see right here, I've drawn a double bond. And what this creates is all of this space where there would have been hydrogens in the saturated version, but now there's not. So this is just space. And because there's all that space, these are a little more floaty. They're not packed quite as tightly. And so the more double bonds you have or triple bonds, the more unsaturated your fats are going to be. So these are things like olive oil, omega 3s, um, and these are typically the oils and the fats that people say are good for you, that you should eat them. And they're typically liquids at room temp. Because not packed as tightly. Now, this double bond that I circled up here is called a cis double bond because both of the hydrogens, I'm going to highlight them. Both of these hydrogens right here are on the same side. So we'll talk about that in a second. But since they're both lined up together next to the double bond, it's called a cis double bond. Now we're going to come over here. We're going to look at trans fats. So trans fats also have a double bond. Here it is. But instead of the hydrogens being on the same side, now you have one hydrogen here and one hydrogen here. And what that creates is this sort of diagonal line right there. And this allows them to pack a little more tightly because there's not quite as much space as there was in the unsaturated version. So this is called a trans double bond. Now, the deal with trans fats is basically, if you've ever had natural peanut butter, you understand this, that when you have an unsaturated fat, it separates. And if you ever eaten natural peanut butter, you've experienced this. You have to stir the peanut butter to mix the oils with the peanuts every time you want to eat it. People are like, that's inconvenient and not pleasant. So what scientists did is they created trans fats by just moving these hydrogens around. They were able to create something that was a little more solid than it was before. And people were like, yay, we like this. Now, the bad news is that trans fats prevent your body from being able to pick up bad cholesterols that you eat and move them to your liver to be processed. So what ends up happening is if you eat a lot of trans fats and saturated fats, is your body starts to build up those fats in your arteries, which can lead to pretty bad heart disease, cardiovascular disease, heart attacks, and strokes. So a lot of things now will actually be labeled as like trans fat free or no trans fats because people have started to figure out that they're not super good for you, but they're still out there. So examples of trans fats, things that are like margarine, um, I'll just leave it at that. But like the processed peanut butters is also a good example. And these will be solids, but they're not like typically meant to be. They've been like edited to be that way. So I'll just make a note here that these are not good to eat. These guys, the saturated ones, they are typically good fats. Okay, that's it for lipids. Now we're moving on to proteins. All right, so proteins are kind of like the powerhouses of the biomolecules. They're super important. Everything that is the way that you look and the things that you're able to do is because of a protein. So like your skin color is because of a protein, the texture of your hair, the color of your hair, um, your nails are proteins, your like the ability of your muscle fibers to move is proteins. Everything is a protein. Proteins, the elements are oxygen, carbon, hydrogen, and nitrogen. And I'll just say like plus some others, which I'll tell you why in a second. But those are the main ones. Your these guys are the main ones. Proteins are made up of amino acids. That's the monomer. And we'll talk about them for a second over here. So there are 20 different amino acids, and that's it. And all those 20 amino acids just recombine in different ways to create all the proteins that exist, which is like millions and millions of proteins. Every amino acid looks like this. So what you have here is a group on the left with a nitrogen and two hydrogens, and this is called an amino group. And that's how they get their names, amino acid. Over here, this thing is called a carboxyl group. And what happens is the amino groups link to the carboxyl groups to make these long chains, which we'll talk about in a second. This thing, R, is called a side chain. There are 20 different ones. And the side chain is what really gives the amino acid its special qualities. So it's kind of the most important part of the amino acid. Here's a picture of all the 20 different amino acids down here, and you can just see the different side chains and how different they really are. All of these side chains give the amino acids their special properties. So these ones up at the top are electrically charged. These ones are polar. The ones in green are nonpolar. It says hydrophobic, which is nonpolar. You do not have to memorize these amino acids. I'm just showing them to you so you can kind of see the differences between them. And they're going to be important for when the protein folds. So those are your amino acids. Okay, let's go back over here and talk about functions. So the function of proteins is that when you eat them, they get broken down by your body, and then the amino acids from the proteins you ate are used to rebuild the proteins that your body actually needs to survive. So basically, you kind of are whatever you ate because the amino acids that were in the protein you ate are being reformed into the proteins that are you, which is crazy and like pretty cool to think about. One thing that I just need to write that always happens is that proteins do not give you proteins not equal to energy. Okay, they are not an energy source. The energy sources are carbs and lipids. What they do is they break down your food. Those are called enzymes. They communicate with your cells. I'll say with other cells. They let things in and out of your cells. They protect you from disease. And there's so many other things that they do too that we'll talk about throughout the year, but this is just a few of them. The ones that protect you from disease are antibodies. Oh, and then I will put this on here too, but they're also hormones. For example, insulin is a protein hormone. Okay, so now let's talk about how proteins fold because this is super important. When we talked about um, carbs, I talked about how structure is really, really important for the protein or the carb doing its job. That's true about proteins too, maybe even more. So I'm going to say over here that this is very important because if shape is wrong, protein doesn't work. It can't do its job. And then often, if people's proteins are incorrectly folding, it's because they have a genetic disorder. And we'll talk more about that when we get to genetics. So, first of all, the first step is called the primary structure. Primary means first. So, this is just the string of amino acids linked together. So, on this little diagram, each dot would be an amino acid, and the bond in between would be a covalent bond, which in this case is called a peptide bond because it's a link in between proteins, and another word for protein is peptide. And that's the primary structure. It's really important that this structure is right because later on it will affect the fold if it's wrong because of those R side chains on each amino acid. Um, they'll interact in the incorrect way if the order here isn't right. All right. So the secondary structure is when that chain of amino acids folds up in a specific way. So this is caused by hydrogen bonds, which we talked about last week. So hydrogen bonds form in between the amino group and the carboxyl group of amino acids that are not directly next to each other. That are not, I'll say, adjacent. So there's two shapes that form when this happens in the secondary structure. The first one is called an alpha helix, which is basically a spiral, and the second one is a beta sheet. So there are hydrogen bonds that are causing these folds to occur. Within one protein, you can have an alpha helix and a beta sheet, and you can have several alpha helixes. I'll show you a picture in just a second that will help. That's the secondary structure. The tertiary structure, tertiary means third, is a 3D shape, forms because of specific chemical bonds between R groups. The R groups were those things that I showed you a picture of before. They all have those special properties. So they could be ionic bonds, they could be hydrogen bonds, more hydrogen bonds. They could even be covalent bonds. So different bonds are going to pull different R chains together, and that's going to fold up the protein into a very specific 3D shape that allows it to do the job. Now, the last level of protein folding is called the quaternary structure. Quaternary means fourth. And this only happens in some proteins, say only in large proteins. But what happens is that many protein subunits that have been folded join together to make large protein. An example of this is hemoglobin, which is the protein that carries your oxygen around your blood, but it is actually 4 protein subunits that link together to make a larger protein. Okay, I'm going to show you a picture of this because I think it will help. So over here is a diagram in your OneNote that kind of shows you. So the primary structure is these little circles that are joined together using peptide bonds. The 3 letters are the names of the amino acids, which you don't have to memorize. Then, using hydrogen bonds, they curl up into an alpha helix or a beta sheet. And then in the tertiary structure, the R groups interact to form this very specific 3D shape. And what's happening here is the red lines represent beta sheets, the green curlies represent alpha helixes. The gray lines in between are still part of the protein, but they just didn't form alpha helixes or beta sheets. They're interacting because of those R groups. And then finally, you might have a quaternary structure where you have different molecules interacting. This one is hemoglobin. So each of the different colored chains are different protein chains, and they're interacting together. That's protein folding, and that's the last thing I'm going to say about protein. Our last biomolecule is nucleic acids. It's short. So nucleic acids, the elements are carbon, hydrogen, nitrogen, oxygen, and phosphorus. So almost all of those ones that last week we talked about were found in all living things. The functions are there's only 2 examples. They're DNA and RNA. The monomers of nucleic acids are called nucleotides. This is a very simplified diagram that shows a nucleotide. So your nucleotides are made up of phosphorus, they're called a phosphate group, which is covalently bonded to a sugar. Depending on whether it's DNA or RNA, the sugar is either ribose or deoxyribose. If it's deoxyribose, it's DNA. That's why it's a D. And if it's ribose, it's RNA. The last thing is a nitrogenous base. This is where the nitrogen comes in. There are 4 nitrogenous bases found in DNA, and then there's an extra one that's only found in RNA. And they are called thymine, which is a T, so how we refer to it, adenine, which is A. These bond together. Cytosine, which is C. And guanine, which is G. These bond together in DNA. Then there's also uracil. I think that's actually supposed to be an A. Uracil is U. This is only in RNA. We're going to talk about nucleic acids a lot more when we get to our unit on DNA. So for now, I'm just going to leave it at that. Okay, guys, this was a lot of information. Let me know if you have any questions about any of this stuff. But these are the 4 biomolecules: carbs, lipids, proteins, and nucleic acids. And that's it for this video. Thanks for watching. Bye.